last session of 2 days international webinar on COVID-19.
Dr. Chakraborty, please close your screen sharing now.
Okay.
Then you set it. Okay.
In this session, we have two successive lectures, one by Dr. Tanmoy Chakraborty from University of Exeter, UK, and the last one is from Dr. Gargi Bandyopadhyay from USA. So without further delay, I would like to request Dr. Somit Majumdar, the organizing secretary of this webinar, to deliver the kind introduction of the speaker.
Okay, thank you, Dr. Bhadro. And good evening to all of the participants who are eagerly listening throughout. In this session, as told by Dr. Bhadro, we have two lectures, and I have been told to introduce Dr. Tanmoy Chakraborty. So before that, let me first introduce myself. I am Dr. Somit Majumdar, working as an assistant professor, Department of Chemistry, Bhorak Ganguly College. Today, we are pleased to have Dr. Chakraborty with us. Hello, Dr. Chakraborty.
Hello. Hi.
Dr. Chakraborty got his PhD degree from Jain CSR under the mentorship of Professor Kaustubh Sharma. He is the recipient of prestigious Marie Curie ITN Fellowship when he was pursuing his postdoctoral research with Professor Attila Gasser, Department of Microbiology, University of Szeged, Hungary. He is now working as a postdoctoral fellow with Dr. Duncan Wilson in the Department of Medical Mycology of University of Exeter, United Kingdom. His current research focus is the regulation of zinc homeostasis in human fungal pathogen Candida albicans. Now I would like to over Dr. Bhadro.
As I have told earlier, it is our deliberate step to present different the speakers from different field. So as you already heard from Dr. Majumdar that Dr. Chakraborty is the expertise in virology and immunology. So listen to him from his perspective. With this, I would like to request our Vice Principal, Dr. Sanjit Kumar Das, to formally welcome Dr. Chakraborty on behalf of our college.
Thanks, Dr. Bhadro. Now I take the privilege to welcome and invite Dr. Tanmoy Chakraborty, postdoctoral fellow, MRC Center for Medical Mycology, University of Exeter, UK, for his presentation. So please, Dr. Chakraborty, now it's time for you.
Can everybody see my screen properly and everybody can listen me?
Yes, it is okay now.
Okay.
Go ahead, please.
So good evening, everyone, and thank you all the panelists for the kind introduction. So I basically works with fungal immunology field. I'm not an expert in virology, but I try to give my perspective on the recent epidemic of COVID-19 from my knowledge and whatever study or knowledge I could gather. So as everybody knows by now that this is a global pandemic which is caused by a family of coronavirus, and this is a false colored electron microscopic picture of the coronavirus. I think everybody has seen this before. So okay. So coronavirus. So when people say that the coronavirus we are getting infected with the coronavirus, it's not actually the coronavirus. We should not say that, because it's the family of virus which contains many different kind of virus, and COVID-19 is one of them. So coronaviruses are actually RNA viruses which can cause diseases mostly in mammals and birds, and they cause respiratory tract infection in mammals from mild to very severe. And this mild infection are caused is very similar to the common cold that we got every year in the seasonal, which is called also seasonal flu, which is caused by rhinoviruses. And the three main deadly coronaviruses that has emerged in the last 2 decades are SARS, MERS, and COVID-19. The recently on the COVID-19. So there are different classes of coronaviruses exist in the environment. and they are divided into 4 main classes like alpha coronaviruses, beta coronaviruses, and gamma coronaviruses. So these 4, you can see in the screen, these 4 are the coronaviruses that cause like mild symptoms among mammals, such as like runny nose and like common flu, common colds, this kind of infection. But these 3, the lower 3 viruses, are the deadliest pathogenic viruses that can cause severe respiratory syndrome in humans. So how the name coronavirus came, or how the scientists give the name like COVID-19. So there is an international committee of taxonomy of viruses who, the people in the scientists there, they decide how to name the viruses. So this coronavirus name comes from the co come from the corona, the VI from virus, D for disease, and 19, the year where the infections first started. So that's how they name the new virus as COVID-19. And as I showed you before, previous slide, so these are the 3 main deadliest coronaviruses that has emerged in last 2 decades, and which cause a global pandemic, and which also has a really high mortality rate, and also has the economic collapse for our society. So these 3 viruses are the MERS-CoV, which happened in 2012. Then came in 2003 the SARS-CoV-1. This is the previous version of the SARS-CoV-2 virus, and the recent one, as everybody know, the main focus of our talk is the SARS-CoV-2, which also called COVID-19, which originated in 2019. So I think everybody is very familiar with this new this computer-generated 3D structure of the coronavirus, but I'll still like to give a brief introduction about the virus structure and how they infect the cells and how they replicate. So this is the computer-generated model of the human COVID-19. As you can see, these are the spike protein, the S protein, and the genetic material is the RNA in the red color. And there are more other proteins like yellow, the E protein, and this is the lipid membrane, which actually makes the shell of the coronavirus. And when everybody is saying that you have to wash your hands with soap and detergent, so this soap and detergent actually disintegrate this lipid membrane structure, which helps to kill the virus. And the genome of coronavirus is a RNA genome. So it does not have a DNA like human or eukaryotic cell, but it has a RNA genome. And the RNA genome actually codes for different proteins that are the part of the coronavirus. As you can see, there are proteins like the protease protein, the S protein, which actually makes the spikes of the coronavirus. And the name corona actually come from this corona-like structure, which is made from the spike protein. And they have endonucleases, which is also very surprising because as you can see in the left, this endonuclease is not common among RNA viruses. These are the different example of RNA viruses like rhinovirus, HIV, Zika, influenza, Ebola. I think most of us have heard about this virus. So this coronavirus is unique that because they have this endoribonuclease. What this nucleus actually does, when the cells makes this new RNA molecule from the existing RNA molecule from the virus, if there is any mistake in the RNA molecule during the process, so this endonuclease can repair the genome of the coronavirus to make a proper similar RNA structure when the virus is replicating. And this is also interesting that this coronavirus family, this COVID-19 virus, has a 30 kb genome, which is not usual in different RNA viruses. So how the cells or the coronaviruses can attach with the human cells. So as we can see, the spike protein actually bind with the ACE2 receptor, which is present in our epithelial cells. So this epithelial cells, this ACE2 molecules are not only present in lung cells, they are also present in our olfactory olfactory epithelial cells, which is present in our nose and eyes and in our throat. So when the doctors started giving the symptoms of coronavirus, as it came into focus, that when we are infected with the coronavirus, the COVID-19, we lost our ability to smell and taste food and different smell. So this is one of the reasons, because this ACE2 receptor is not only present in the lung epithelial cells, but they are also present in our olfactory epithelial cells. So this coronavirus, the S protein, the spike protein actually bind to the ACE2 receptor, and then this protease enzyme, which is present in our epithelial cells, which cut this the stem of this spike protein, and then it engulfs the virus inside the cells. So this is the whole replication cycle of the virus, or the propagation cycle of the coronavirus. So after the binding of the virus to the ACE2 protein and the cutting of the protein from the stem, so the virus enters our cell, and this process of infecting the cells is called endocytosis. So after the cells, after the viral molecule or the viral particle is inside the cells, with the ribosome, which is the host ribosome or human ribosome, it starts translating the RNA. So and makes the different viral proteins, as you can see from this place. These are the spike protein, the N protein, and we have to keep it in mind that the virus does not have their own machinery to do these things. So the ribosome is from the host cells. So when after generation of all the spike protein and the E protein and the other important protein on the endonuclease proteins are there, that they get integrated in the Golgi membrane or the ER membrane, and from this membrane the virus assemble their genome, and the whole assembly is done, and then the virus again comes out of the cells by the means of exocytosis and start infecting other persons or other individuals. So there are different detection strategies came immediately after the pandemic has started. So there are different strategies to actually detect the virus inside our body. So the most commonly used or most robust strategy, or to very positive strategies, to use the real-time PCR. But there are many other strategies like this antibody-based strategies. I think most of us are hearing about this antibody-based strategies and the whole RNA extraction, whole RNA sequencing strategy. But these are the antibody-based strategy is the most easily available strategy, and which can be done in very small time span. But the most robust strategy is the real-time PCR, which can detect the viral RNA inside our blood. So how does this real-time PCR work? So we take the sample from the nose or nose swab or mouth swab, and then we extract the RNA from the viral particle, and then we from the RNA we make the DNA, and then this DNA can be identified with different dye and probe and with the polymerase reaction. That's why we call it the PCR reaction or polymerase chain reaction, or real-time polymerase chain reaction, because when you run the samples in the real-time machine, you can see in the real time the positive and the negative samples. So this is the most valuable strategy to detect RNA viruses or detect COVID-19 virus in our body. So as Debabrata asked me to actually focus on the treatment strategy or prevention strategy for COVID-19. So I'll go through some of the the antiviral drug strategies, and the vaccine strategy, which is the main focus of my talk today. So, so there are different antiviral drugs in trial still now. We cannot say that all are like confirmed to cure the virus completely. So there are various kinds of drugs scientists and doctors are trying to actually get rid of the virus or kill the virus. So the first class of drug which actually blocks the ACE receptor and this protease enzyme, which is present in our epithelial cell, to actually block the binding of the virus with the ACE2 protein. So if the drug blocks this binding, actually then the virus cannot bind to the ACE2 cells and cannot infect us. So these are one class of drug, this chemostat, which is actually blocking this interaction between the viral spike protein and the ACE2 receptor of the human cell. So the other kind of drug is which actually blocks the protease enzyme inside our body, which actually helps to process the viral protein inside our cells. So this kind of drug is lopanavir or darunavir, but these are not exactly healthy, because this can also mistarget the host protein or human protein, which can be deleterious for the host. The third is it's like an anti-inflammatory drug, which is very common for very different viral infection. So this kind of drugs actually blocks the cytokine molecules. So the cytokine molecules are actually signaling molecules inside our body. So when the virus infect our body, the cells or the immune cells start producing different signaling molecules. So cytokine or interleukin-6 is one of these molecules, and these drugs actually try to block this IL-6 receptor, or actually try to block the IL-6 production inside our cell. The most of the patient which goes to the or which has to be taken to the what you call the ICU patient, mainly the ICU patient who has to keep the ventilator support because of this reaction. Because if this uncontrollable reaction happens inside the body after the viral infection, that too much of interleukin molecule can damage your lungs, and you get this problem of hyperventilating. So the hypoventilating. So these drugs try to block this immune system, the immune reaction inside the cells when the virus infects the human host. And finally, there are another drugs, which is flavivir or remdesivir, which is coming in focus in recent days. So they try to block the viral RNA polymerase, which actually makes the RNA, the virus, which actually replicate the viral RNA and makes more RNA to generate new viruses. This drug has been successfully used for HIV and other RNA viruses, and these are very specific because the human host does not have the RNA dependent RNA polymerase, which is required to replicate the viral RNA to make more viral RNA. So this is one of the most important drug for this COVID-19 patients or COVID-19 infection. There is another therapy which is currently in use in many places, and some cases it has some successful outcome. These are this covalescent plasma therapy, and I think most of us are very familiar with the situation, but I just go through it in very simple terms. So in this covalescent plasma therapy, what we do, we take the blood from the patient who are recovered from the RNA, the COVID-19 symptoms, and we take their blood. And we try to isolate the antibodies, which has been generated in the the patient, and we try to use this antibody, take the antibody, and give it to the infected patient. So this antibody from the infected patient can kill the virus in the next patient. So this is also used. This has been also used very successfully, but the drawback of this plasma therapy is that this cannot give you a long-term immunity, because these are only antibody molecules that has been generated in the other patient, but not inside your body. So it can give you the memory reaction that is required, that is generally used by the vaccine. But still, it has been used in some cases successfully, but it is not overall. Not every country is using this therapy till now. So the best way to tackle a viral infection is to generate or make a viral vaccine, which is very successfully used in many other viral infections such as smallpox and polio and so on. So to generate vaccine strategy for COVID-19, many different companies or many different institutes are trying to use different strategy to generate antiviral vaccine for COVID-19. These are like this. These are the 5 or 6 main strategy, which I'll go through in detail from the next slide. So the generation of a vaccine requires few steps, which takes a longer time. So when the viral infection started, I got question from many of my friends and family that when the vaccine will be available to the common people. So these are the timeline for generating a normal viral vaccine. So as you can see, there are different stages, which is called preclinical stage, clinical testing, and monitoring. So in the preclinical state, in preclinical testing, we use different model system like mouse, ferret, or rhesus monkeys to test the vaccine in this animal model. And then if we see a positive result in the animal model, then we go to the clinical testing. Now this clinical testing has also different stages. So these are the phase 1, phase 2, and phase 3. In phase 1 stage, we actually test a very small number of volunteer population and see if how the viral vaccine is working. If it is successful in the phase 1 stage, then we go to the phase 2 stage, where we take more larger number of sample, a larger number of volunteer to use the vaccine and see their immune reaction or how the vaccine is working on them. And finally, we go to the phase 3 stage, where you take a larger population and infect like thousands or more than 1,000 people and see the effectiveness of the vaccine. As you can see, in the normal cases, it takes at least 10 to 15 years. But because the COVID-19 is a very serious situation, or so many companies are and the institutes are trying to do the same task in very short amount of time span. And finally, the phase 4, this is the mass vaccination stage, where we try to generate a huge quantity of vaccine and try to give to the normal people. So there are actually 160, as far my knowledge, there are at least 164 candidate vaccines which are on pipeline to give to give to the common people to generate the anti-vaccine strategy, antivirus strategy. And I'll talk about 5 of them. The most promising one and which is in the phase 2 of its clinical trial until now, is by a company called Moderna. So what they're doing, they are generating the mRNA vaccine for COVID-19. So what is the strategy here? So as I showed you in the previous slide, the mRNA code for many different proteins, like the spike protein, the R protein, the polymerase protein, and everything. So what this company is doing, they are taking only the mRNA portion of the viral genome, which only codes for the spike protein for the virus, and they are injecting this mRNA particle in the host cells, and the RNA will generate the spike protein after the translation, and our immune system will try to generate antibody against this only the spike protein and see. And when the next time the virus is inside the cells, or when we are getting infected with the virus in the next phase, this antibody can give us protection against the virus. But like all the vaccine and all the strategies, there are risk factors with every vaccine strategy has their pros and cons. So the RNA vaccine strategy has different risk factors, which are like they are very unstable, and sometimes you can get off-target response. And when the protein is generated, the viral protein is generated inside cells, it cannot be like proper protein, or it has some mutation which can hinder the actual immune response against this protein. And we require specialized administration, like we have to inject direct mRNA injection or something to insert the mRNA inside our body. So these are some risk factors associated with this mRNA vaccine. And this vaccine is in its phase 2 of its clinical trial. The next one is which is mainly generated or in collaboration with the University of Oxford and AstraZeneca in UK. So in this strategy, what they are doing is very similar to the previous one. So they again take the gene encode, the viral gene which encode the S protein for the COVID-19, and they put the gene inside in another virus which is called a chimpanzee adenovirus. But this virus cannot infect the host cells, so they are modified virus, and they put the RNA portion of the COVID-19 which can code the spike protein, and fuse them or insert this RNA in the adenovirus genome. Sorry, in the empty adenovirus, and then we infect humans with the adenovirus, with this adenovirus which contain the SARS-CoV-2 mRNA. And when the cells enters in the body, again this RNA will make the spike protein, and our immune system can generate antibody against the spike protein. And then further, when we get reinfected with the virus, we can have a immunity against the COVID-19. The other problem, the risk factor with this virus, with this strategy, is this genome can sometimes integrate in our own chromosome, which can give cancer manifestation. And if we already have pre-existing vector immunity, such as if we have infected before with the adenovirus somehow, we can have the antibody against those viruses, and which can make the situation complicated. And this vaccine is our phase 2 or phase 3 trial, and it's going to be, so as per my knowledge, in June, they are starting the phase 3 trial of this vaccine. The other company, which is called BioNTech, they are also taking the same mRNA vaccine strategy, which is very similar to the previous two strategy. But in this case, they are actually trying to take different combination of the mRNA of the COVID-19. They are not only taking the spike protein encoding genome, but the other proteins such as the RNA polymerase or the nucleus, and try to insert those this different RNA molecule in a lipid nanoparticle, and then inject this combination of RNAs inside the human, and try to generate the vaccine. And so in this case, the human not only will generate antibody against the spike protein, they can generate antibodies against the other different proteins like the RNA polymerase or the endonuclease. And when we next time get infected with the virus, we can have antibody or protective immunity against the RNA virus. But again, this also has some risk factors associated with this strategy, which are sometimes they can give atypical antigen processing. So when the mRNA is inside the body, the mRNA will generate the viral protein, and the protein production cannot be always perfect. So that we can get a atypical immune response against those proteins, which can be complicated for the patient. And again, it also requires specialized administration, because we need to generate this lipid nanoparticle to deliver the RNA molecule inside our body. And this strategy is also in the phase 2 clinical trial until June. This month, they are starting the clinical trial in the phase 2 clinical trial. So until now, so all the vaccine that I was discussing is about the RNA vaccine strategy, but we can also do a DNA vaccine strategy. How? So here, so we take the RNA of the coronavirus, and again they are trying to take the spike protein sequence encoding portion of the genome of the RNA virus, and they reverse transcribe the RNA molecule and generate a DNA which can code the spike protein when inside our body. So they do it in vitro or artificially, outside the body. So they generate the DNA from the RNA molecule of the COVID-19, and that DNA only contains the gene of the spike protein from the RNA virus. And with a specialized electroporation strategy, they can we can insert this whole DNA construct inside the nucleus, and then the DNA will produce the RNA, and the RNA can produce again the spike protein. And again, the strategy is very similar to the RNA protein, similar to the RNA vaccines, because the RNA can generate the spike protein, and our immune system start generating antibody against the spike protein, and that's how we get the antiviral immunity inside our body. This strategy is also used by one Indian company, which is called Serum Institute of India, with the foreign one, the Codagenix, and they have a collaboration to generate this vaccine. And this is also in the phase 2 trial in the June. But again, all the other vaccine strategy, this also has risk factor associated with, because the DNA can integrate in our genome, which can cause oncogenesis or cancer generation, and it can also have off-target response. And this is because this DNA has to go inside our nucleus, cell's nucleus. So we need a special administration strategy like electroporation, or we call it gene gun sometimes, to administer the DNA inside the nucleus, which is not always easily available in poor countries or in all different setups. The next vaccine strategy is called the attenuated viral vaccine strategy. This is also a very common vaccine strategy, where we try to use some virus which is weakened or which is inactivated. The weakened virus means what they do. So the virus is intact. In some cases, the virus is intact, but they cannot generate the disease when it's inside our body. So So there are two strategies. One is you can inactivate the virus by using different chemicals such as formaldehyde or heat and make them so they are dead. They are simply dead viruses which cannot replicate inside their body when it get integrated. And this viral strategy, so in this case, it is more simpler compared to the RNA or DNA vaccine strategy, because here you kill the virus and then use the virus as a vaccine strategy to inside our body. So, but again, this strategy also has the drawbacks, because this need a very extensive safety testing, because when you are administering the vaccine inside our body, we cannot have a live virus. So we need to be very careful when we are injecting this inactivated virus or weakened virus inside our body. And this is another challenge for this kind of vaccine, is we have to manufacture a lot more number of viruses to use it as a vaccine and inject in our body. So this is also not very easy to generate viruses, because virus. So as we know, viruses are inanimate particles. So we call it particles. They are not live. So we need host cells to generate virus, and making a lot of virus is not an easy task for generating a vaccine. And so finally, there's another strategy which is called the protein subunit vaccine for COVID-19. So last few strategies is mainly based on the genetic material of the virus, but in this case we are using the protein molecule of the virus, such as the S protein or the M protein. So here again we have to make a lot of virus to get this S protein and inject directly the protein inside our body to make to generate the antiviral immune responses in our body. And another one is a virus-like particle, where you have only the shell of the virus, the main structure of the virus, which is called virion, but we don't have the RNA or RNA inside the virus, so that when the virus gets inside our body, it cannot replicate or multiply inside our body, but they have only, because they don't have the genetic material, so we have only the viral particle, and our immune system can generate antibodies against this the viral particle, only the viral particle. So it's a good strategy to generate vaccine. But again, it has been shown that they have poor immunogenicity. I mean, the immune response is not that strong compared to the RNA or DNA viruses. And when we have to use this viral particle or the protein structure, subunits for this vaccine, we need to require adjuvants, which are generally like metal, mainly aluminum. We use aluminum to make this adjuvant, and we use aluminum to make this vaccine to insert this viral particles inside their body, and it has a limited global capacity. So another question which was coming, or which the study was coming during this whole pandemic study, and I was lucky to listen to a talk by one of the immunologists who is working in our field, like anti-candida immunity. So does BCG vaccination has any protective immunity against novel COVID-19? So so there are 2-3 contradicting studies regarding this BCG vaccination and anti-COVID-19 immunity. So they showed that in many cases, mostly in mouse model, when the mouse is given the BCG vaccine in different doses, they can generate antiviral immunity against some viruses, like influenza, this human papilloma virus, and human papilloma virus, mainly these two. And in some cases, they give some immunity against other bacterial pathogens also and fungal pathogens also. But we still don't know how the whole mechanism is working. And in some cases, it has been shown that this BCG vaccination gives protective immunity, but there are 2 studies from Israel where they have shown, because they also have the BCG vaccination among the population, but they don't show any protective immunity against the COVID-19. So we need more research to understand this whole BCG vaccination strategy is working against COVID-19 or not. And another strategy, which is called herd immunity, we heard from many reports. Sorry. So we heard from many different places many times that herd immunity be used to give the protective immunity to the larger number of population. So what is herd immunity? It is like you It's an indirect protection from infection conferred to susceptible individual when a sufficiently large proportion of immune individuals exist in the population. So it's a very simple strategy. So we generate our the immune system against the virus spontaneously just by getting contact with the infected individual, and that's how the whole immunity goes to the overall population, and then we don't need any vaccine and we don't need any protective strategy to generate the antiviral immunity. But the most robustly used herd immunity strategy was in Sweden, in the Scandinavian country, but they have failed very badly because the death per million population is very high in Sweden. As you can see, these are the 4 Scandinavian countries, and only Sweden started using this or started following the herd immunity strategy. And as you can see, their death rate is very high compared to the other 3 Scandinavian countries where they follow the normal protocol of lockdown and contact inhibition and social distancing and so on. So finally, we can see that this herd immunity strategy is not working in case of COVID-19. So so as you can see from most of the vaccine, they are mostly in phase 2 or phase 3 trials of phase 2 and phase 3 trials are going on. So until the vaccine is ready, we still need to follow this common preventive measure or common steps to actually make ourselves safe from this infection, and which is like wash your hand, avoid touching your nose and mouth and eyes, and try to avoid crowded places, and the normal that we are always hearing from all the government and all the agencies to keep safe us from the COVID-19 infection. And with this, I like to thank you everyone for listening, and if you have any question, I tried to answer.
Dr. Bhadro.
The time is fine, right?
Yeah.
Dr.
Bhadro,
please
go ahead.
With the question answer session. Dr. Majumdar, are you?
Hello.
You carry on with the question answer session.
Okay. So the participant, if you have any question, please, you can. Hello. Is there anybody who have any question?
Questions are already in the chat box.
Okay.
You follow question answer box.
Okay.
I can see some questions.
Yes, you can see also, Dr. Chatterjee. Chakraborty, sorry.
Yeah, one question is from Arnav Banerjee. What would be the possible effects if 2 antiviral drugs, favipiravir and imipenetrovar, are used simultaneously in patients?
So yeah, they are also trying to use this combination of drugs to inhibit like viral RNA polymerase or the spike protein or the SC2 inhibitor, but we are not still sure how this combination is effective. In some patients, they are curing, they are getting fine, but in some patients, it's not effective. So the whole combination drug therapy is still not properly understood yet. So as far as I can say till now.
So again, the one question is from Arnav Banerjee. Why are not using CRISPR
CRISPR strategy,
for viral inhibition, which can potentially destroy the SARS-CoV-2 sequence?
Yeah. So the CRISPR strategy is also the scientists are trying to standardize the CRISPR strategy to actually target the viral RNA. It's also going on, but it's also a very good strategy if we can use it properly. Because the CRISPR strategy also has some drawbacks, because the CRISPR Cas9 protein, the Cas9 protein is actually sometimes generate immunity, again, anti-Cas9 immunity generated inside our body, so which can make the situation more complicated. So that's why this CRISPR strategy is not like frequently used for many human diseases. People are trying to do it, but it's not in use yet. But they are also trying to use this CRISPR strategy to actually kill the virus, I would say, or or destroy the mRNA, because it's CRISPR actually target the genome of the virus, not the protein or the other part of the virus. So it's in use. I mean, it's not in use, sorry, it's in trial, or people are trying to generate the strategy to use against the virus.
So no other question. I think another question is from Arnav Banerjee, but other participants are not asking any question. So this is a question from Arnav Banerjee. The FDA in a drug safety communication said that hydroxychloroquine has been given emergency use authorization for the treatment of patients who have tested positive with coronavirus, but it has been linked with serious and potentially life-threatening cardiac arrhythmias. So can it be a fruitful strategy to fight COVID-19 where it can initiate a deadly cardiac problem?
So, now the problem is most of the places or most of the countries, the FDA actually disapproved the chloroquine or hydroxychloroquine treatment against the COVID-19 patient, because it actually is not helping to reduce the mortality rate among COVID-19 patients. So there is still research going on, because this chloroquine or hydroxychloroquine has a lot of negative side effects inside our body. So this is still not confirmed that we should use it or not use it. It's still in a contradictory study. Different contradictory studies are going on. So I think we need more time to actually just say or ask the doctor to use chloroquine to treat the COVID-19 patient.
Okay, another question from our colleague, Dr. Subharti Pal. Is plasma therapy under active consideration in UK yet?
I think in UK, they are using the plasma therapy, and they are actually, so this I did not discuss in my talk. So they are using recently they are using a steroidal therapy to reduce that mortality rate among this COVID-19 patient, which is, they are saying, which is very successful. So the steroid drugs are actually anti-inflammatory drugs, which doctors are very commonly used in the ICU patient. So, but the plasma therapy, yes, in UK also they are using, and they have some success in that also. This steroidal drug, it just came last week. The news just came last week, and they show that if we use this steroidal drug, so you can actually, so 1 from 10 ICU patient can be recovered, or 2 from 10 ICU patient can be recovered using this steroidal drug.
So another question is from Ishita Chatterjee. She is asking, does the virus undergo mutation? If so, then does the virus change its symptoms?
Yeah, it is also possible, because the viral RNA, when the cells makes new viral RNA, so we can have mutation during the replication process. But the COVID-19 is interesting in the case because they have this endonuclease which can actually repair the RNA molecule during the replication. So from my knowledge, I would think the mutation rate would be very lower compared to the other RNA viruses, because they don't have this endonuclease which can actually correct the mutation in the RNA molecule. So I would think that the mutation rate will be less. Yes, and definitely if there is mutation, there will be structural variation in the viral. The viral capsid, the spike protein, which can also makes this either more virulent or less virulent, depending on the situation. Yeah, it is possible. Yeah.
There are so many questions coming. Is it okay, Dr. Chakraborty, for you?
Yes, it's fine.
Okay.
I mean, if the timing is permitted for the next talk, I think it's fine.
Okay. So I think we can entertain another question. So Amit Mandal, does sunlight can kill COVID-19 virus?
I am not sure about this, so I cannot answer this because I'm not sure about it till now. Yeah, so they say this UV sterilization is working in some places. In New York, in big cities, they are actually cleaning the metro coaches or the rail coaches with the UV light, and the sunlight has UV light, but I don't know how effective it can be for this virus.
So just one is my question is one question there from my side, that we know that normal detergents is okay. When we are doing hand wash, so like that, vegetables from market. So some people are sanitizing, but just by dipping this vegetable into the detergent. What do you think? This is safe, or it is very much?
I think if the detergent goes inside your body, I don't think it's a very good option. And I'm not sure that how much the virus can survive in a plant leaf or inanimate object. So there are some research they can survive for some time in a inanimate subjects like table chair or something, but the study is still inconclusive. And the vegetable part, I don't think we need to do it and dip the vegetable inside the detergent solution, at least from my opinion or from my knowledge.
Dr. Chakraborty, I have one question.
Yes, please.
Is tree plantation reduces the chance of spreading coronaviruses?
Pardon me, please. I could not.
Tree plantation.
Tree plantation and reduction of corona. I don't know. I don't know. I don't know.
Thank you very much. We have some participants with us who raise your hands. Should we take them? Dr. Bhadra or Majumdar?
We have time until 6 p.m. before Dr. Bandyopadhyay will start. So let's take 3-4 questions till then.
So one of our participants is there, Vaishnav Wak. Let we call him to talk, ask you questions.
I have a question.
Yes, Dr. Wag, please introduce yourself.
Yes, I am Vaishnav Wag. Hello.
Please introduce yourself.
I am Vaishnav Wag from National Institute of Virology, Pune.
Yeah, please ask your question.
I have a question, like since the PPT was about mechanism of entry of virus into the host cell, there we are using TMPR SS2 for spike protein priming, right?
Yes.
So does that give us any idea about drug formulation that can target this site?
Yeah, we can. I mean, people can actually, or scientists can target those molecules actually, which can actually block that interaction between the virus and the receptor.
So there was so there was the study which was done by done in China, where they showed that this drug doesn't, the drug which was targeted against TMPR SS2 receptors, it doesn't show any side effects as such. So why isn't this thing taken into consideration now for developing a vaccine or something like that?
So you have to remember, you have to keep it in mind that drugs is different and the vaccine is different. So drug is targeting the binding of the virus with the receptor, but the vaccine is trying to give you the lifelong immunity inside your body. So drug is like a preventive therapy, like normal other infection. But I think if it's working, I think people are, I hope the people are taking it in consideration for further use in patients who are in hospitalized patients. But it's a strategy, yeah.
Another participant is there, Dr. Siddhartha Chatterjee, I think. Dr. Chatterjee, are you getting us?
Yeah, I'm with you.
Please.
Okay, am I audible? Okay.
Yes. You are audible, yes.
Please place your question.
Hello, am I audible?
Yeah.
Yes.
Please.
Okay, sir, I just want to ask one question that, from online newspaper, I got this news that one medicine, has been launched by Cipla company, and the medicine name is Cipremine, which can be used for killing coronavirus. So, do you have any information on this matter?
As As I know, this is also target this RNA-dependent RNA polymerase of the virus, but I'm not sure how effective is it, because I know it has been, there people are using, or the doctors are using it, but I don't know how effective it is. But yeah, I heard it. So I think it's also targeting the RNA.
Actually, many medicines name in today on newspaper or on social media, it is coming. So I just don't know that these news are true or not. And for this reason, I'm just asking you.
So I heard about this particular drug, but I'm not sure how effective the drug is.
Dr. Chatterjee, please introduce yourself.
Myself, Dr. Siddhartha Chatterjee, and I'm a teacher or professor of political science from Vivekananda College, Mudhamra.
Okay, thank you very much for your question. Next, we have Prabir Karar with us. Mr. Karar, are you getting us?
Yeah. Hello.
Please introduce yourself.
Hello. Thank you for your nice presentation. Hello.
Please introduce yourself first.
Myself, Dr. Prabir Karar, Associate Professor of Economics, Bhairav Ganguly College.
Okay. Then please place your question, please.
Hello.
Yes,
Hello.
Yes.
Can you hear me? Can you hear me?
Yes.
Okay. I just want to know one thing, that you have talked a lot about the vaccines which are in the pipeline, and you have focused mainly 5 of them, and some of them are in the phase 3 trials. Okay.
Yes.
Now, and also the thing I have come to know from your speech, that in June itself, some third phase trial has started. My simple question is actually, if then after the trial is over in the third phase, so what would be the time that would be taken by these companies or the research institute for this marketing and all these things? Just to say you that a country like ours, India, a large population, and already more than 4 lakhs have been affected, but the recovery rate is high. But if we have some kind of vaccines or something like that, we could prevent the COVID-19 pandemic seriously. So I just want to know that how much time it will take after the trial 3, phase 3 trial, if it is get successful. What is the time?
So if you see the phase 3 trial, when it goes to phase 3 trial, there are 1,200 volunteers. They are giving the vaccine. But the problem is that you have to continuously check the symptoms in the volunteers, if they are developing any side effects, they are having any other problems, so they are actually generating the disease. And so it's a long process. You have to follow up for a long time. You have to follow up the volunteers, how they are feeling, and how is there any side effects among them. So I think it will take another 4 or 5 months to actually completely sure that the vaccine is working and it is giving you a protective immunity. So I think end of this year, or most probably the beginning of next year, we could get a vaccine candidate which can be used in a large scale. And you have to keep it also in mind that this vaccine, we have to generate it in a huge amount to give the whole population. So it's not an easy task. So I think by next year, beginning or end of this year, we could have a vaccine.
Just a minute. There is a rumor around the corner that the second wave will come in sometimes in the winter in India. Is there any chance of second wave of coming at the end of this year?
Yes, there is a chance, because in South Korea and in Japan, in many places, we have seen the patient who they are already getting a new patient, even in South Korea, in China, in China also. In Beijing, we can see there are people getting infected with the viruses. It's not less. The number is not less. So I think India also has the probability of having a second wave of the coronavirus.
But there is a thing that there is no case of regenerating this virus. Suppose a person has already COVID-19 positive, then after he's getting well, he or she could not be affected the virus in the second wave as well.
No, it is not true. It is not true, because there are cases in Japan and South Korea, where a patient is already recovered from the COVID-19 and they got infected again with the coronavirus.
I see.
It is not completely true that you cannot get a second infection if you are infected. But as far my knowledge, the patients are not like severely diseased condition, so they are recovered easily. That's what I say.
Okay.
So, but
okay. Thank you very much.
It's a possibility. Thank you.
Thank you very much. Thank you.
Thank you very much. We have Sulogna Hatai with us. Sulogna Hatai, are you getting us?
Yes, I'm audible.
Please, ma'am,
Yes, yes.
introduce yourself and then ask a question.
Thank you, sir, for your presentation. Firstly, I'm Assistant Professor of Food and Nutrition in THK Jain College, Kolkata, Kashipur. My questions to you is as because you have gone through so many processes that the entire world is working on, how to produce a vaccine, and someone is working on spikes and someone is working on the entire gene. So what, according to you, is India's position to combat or...
Sorry, I can't hear you.
Am I audible?
Yes, yes. Could you repeat the question, please, once more?
Yeah. What, as because you have gone through so many, processes that the entire world is working on. So, what, according to you, is India's position in terms of the progression or the success?
So, as far as I know, this Pune-based company, the Serum Institute, I think the Serum Institute or Serum something. So they are in collaboration with another company, and they are trying to generate another vaccine. So I think India is also in the pipeline, or India is also in the game to generate the vaccine. But
okay,
again,
thank you so much.
Again, with the collaboration with the foreign company.
Okay, thank you so much.
We have Mr. Amit Mondal with us. Mr. Mondal. Mr. Mondal, are you with us?
So I got one question from Subroto Guho. Can herbal medicine is an option as potential drug against coronavirus?
I'm not sure about this because I haven't read any studies or any papers which can actually conclusively tell that the herbal medicine is working against the virus or something like that.
We have Ketan Mukhopadhyay with us. Mr. Mukhopadhyay, are you getting us?
Yes, sir.
Please introduce yourself and ask your question.
Hello, Dr. Tanmay. Thank you so much for your presentation. I'm Ketan.
Hello.
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 Yes.
Okay. So my question is like we heard like Arsenica album 30 is prescribed by so many doctors that can boost our immunity system. So like apart from that, could you please prescribe any food items that can boost our immunity? Because like whatever medicines that doctors are prescribing, that is not reaching to everyone.
So I'm not sure about this the arsenic something you said because the presentation I give is like the study, it's already in online, so there is no study or no paper related to this homeopathic medicine which is acting against the coronavirus, and so I'm not sure how it works or is it actually helping the patients. And the second question, the boosting the immunity, I think we should take a proper diet that we generally use or we eat generally. So I think that is one thing that we can normally do and which can give us a like normal immunity. But yeah, as far as my knowledge, I can say. But about the medicine, I'm not sure, and I don't read any study or I don't have any knowledge about the medicine actually.
So many questions.
Yes.
Let's thank the speaker again for his beautiful deliberation. And from the personal note, Tanmay is my childhood friend. So thank you.
Yeah. Welcome.
So for the we have heading towards the final session of the 2-day webinar. In this session, we have Dr. Gargi Bandyopadhyay. Dr. Bandyopadhyay is now the research scientist at Yeshiva University he has done his postdoctoral research from University of Maryland. He has done the PhD from Jadavpur University. Some of the techniques for molecular biology, she has used DNA sequencing, PCR, RT-PCR, and epigenetics, DNA methylation assay, cDNA synthesis, and many more. I am stumbling to read the words, so I am skipping it quickly. She has the specialization in cell biology and biochemistry, cell culture, immunohistochemistry, and immunofluorescence staining. He has a vast experience of clinical research in vitro translational migration assay, ITEM, with patient FNA, and several research papers in her name, several research projects. So she has a long career of about 12 years of experience in this expertise, and she has our 2 US patent in her name, several publications, as I also already mentioned, in high impact journals. So with this, I would like to request our Vice Principal, Dr. Sanjit Kumar Das, to welcome her on behalf of our college.
Thanks, Dr. Bhadro. Now I have the honor to welcome and invite Dr. Gargi Banerjee, research scientist, Yeshiva University, New York, for her presentation. So now, Dr. Banerjee, please.
Yeah, thank you so much. Can you hear me?
Sure, sure.
Yeah. So you can share, you can see my desktop, right? The presentation?
You have to share the screen.
Please share your screen.
I did that. Now you can see my screen?
No.
Hello?
It is not coming, still not coming.
Still not coming.
You have to share the screen.
I did that.
In front of the window, in the middle of that, you have a green option of share screen. And via that, you have to put the PPT.
Yeah, I did that.
You just close it again.
Okay.
Close the share option and try it again. Better you can try. Turn off your video and try it later.
Share screen. Oh, now it's there.
Yes, now it is okay, I think.
Okay.
Wait.
You can proceed, ma'am.
Yeah, now you can see my screen, right?
Yes, yes.
Sorry for the interruption.
Okay, it's
okay. Okay. So thank you for giving me this opportunity to talk about this current pandemic due to coronavirus and its possible treatment. So before going to today's main topic, I want to describe briefly about my own research field. I am working in Yeshiva University, New York as a research scientist for last 6 years. Basically, my project is breast cancer stem cell project. Our project is a collaboration with Albert Einstein College of Medicine, which is also another leading medical school in New York City. So our group is looking for a unique site which is responsible for the breast cancer metastasis. The unique site is tumor microenvironment of metastasis, TMEM. So what is TMEM? This is the growing primary tumor cells. Some cancer cells are detached from the primary tumor cells and attached to the blood vessels. It's already known that the cancer cells which attach to the blood vessel are more aggressive, and it forms a secondary tumor in the distant organ by the process of metastasis. The cancer cells from the primary tumor enters to the blood vessels. That area form that unique site, which is called tumor microenvironment of metastasis. This site consists of 3 kinds of cells. One is macrophage, another is the endothelial cells, which is part of blood vessels. The third one is the cancer cells. So with the help of these 2 cells, the cancer cells get into the blood vessels and ultimately form the secondary tumor in a distant organ. So my project is actually related to how the stemness of the cancer cells are related to this area, how the breast cancer stem cells are related to this tumor microenvironment of metastasis. To do this, I developed an assay in our lab that is intravasation transendothelial migration assay. So basically, this is just like formation of this TMEM-like situation artificially in the lab. So there is a particular number of HUVEC cells that I cultured and placed on the bottom of a filter. Why HUVEC cell? HUVEC cell is a human endothelial cells, which is actually mimicking these endothelial cells of the blood vessels. So this HUVEC cell, I put it on the bottom side of a filter with a matrigel and let the HUVEC cell grow for 48 hours in a 24-well plate. After 48 hours, then the tumor cells come to our lab. Tumor cells means the patient's FNA sample come to our lab. I colored these patient FNA samples, cancer cells, green color by the green cell tracker dye. And I took the mouse macrophage cell line back instead of original human macrophage cell line, because it's very difficult to isolate human macrophage cell line. So, and this macrophage cell line, I colored red with the real cell tracker dye. There is a particular ratio of the patient FNA sample and the macrophage cell line. That is the 1:4 ratio. So with a mixture of these 2 population of cells, I put it on the top of this filter under which the HUVEC cells layer is already prepared. And I let the assay run for 18 hours. After 18 hours, there are 3 population of cells generated. that is on the one population is on the top of the filter, that is the loading control. The second population of cancer cell is which is the stuck in the HUVEC cell layer, which actually didn't cross the HUVEC cell membrane. And third one, which crossed the HUVEC cell membrane, and it is on the top surface of the 24-well plate. So I collected the only green color cancer cells from these three populations by single cell isolation technique. Is a micromanipulator attached with the fluorescent microscope. This is a pretty tricky method. Anyway, so after the collection of the cancer cells from the three populations, I extracted RNA. The RNA yield was very low because on a good day, I can collect only 10 to 15 cells from each population. So I need to amplify these cells by mRNA amplification technique. So by amplified mRNA, I labeled those with Cy3, Cy5 and hybridized on a chip, microchip. And then finally, I did whole genome microarray. So with a whole genome microarray analysis, it is shown that the breast cancer stem cell marker genes are significantly upregulated in the cells which is cross the HUVEC cell compared to this population, and definitely compared to these top cells. So that means the cells which ultimately cross the blood vessels has more aggressive, has more stem cell property. Among all the stem cell marker gene, 2 or 3 I am mentioning, that is CD133, ALDH1, were very well-known breast cancer stem cell marker gene. So by the studying of these whole genes, that the several pathways came up by studying the microarray data analysis. Currently, I am working with the Wnt pathway, how Wnt pathway is responsible for the progression and promotion of the breast cancer to a distant organ and forming a secondary tumor. So we are using various Wnt pathway blocker. Among them are very unique Wnt pathway blocker antibody. And by blocking this Wnt pathway, this antibody, we have seen that in the animal model, it significantly reduced lung tumor formation or the lung metastasis. So this is briefly about my research work. Now I am going to start today's main topic, that is the current therapeutic approaches for COVID-19 patients.
Sorry to interrupt you, Gargidi.
Yeah, go ahead.
You just keep your PPT in full screen mode before you go into details of therapeutic approach.
Sure, sure. Now you can see my full screen, Devobrato?
Yeah. Please go.
Okay, yeah. So, what is coronavirus disease? Since the first case of this deadly virus was reported in end of 2019, around December, within 2 months, it had become a worldwide pandemic. It's a single-stranded RNA virus with 26 to 32 kilobase pair length. Symptoms. This severe symptom of this respiratory disease, the SARS-CoV-2, varies widely from its asymptomatic to pneumonia kind of life-threatening complications. These are few known symptoms of COVID-19. Those starts from asymptomatic infection to mild without pneumonia, negative RT-PCR by positive antibodies, whereas the complicated with secondary bacterial pneumonia, pneumonia with diphtheria, and the positive real-time PCR or positive virus traces after the recovery. Those are all the possible symptoms of this virus, and which maybe sometime is called the multi-organ failure and ultimately death. The mode of transmission, human-to-human transmission via droplet, is the most active way of the transmit this virus. now various literature in CDC showed that the virus lifespan can depend on the various surface. It depends on the types of surface, because the contaminated hand and contaminated surface also another mode of transmission. So in plastic surface, virus can stay for a particular time. On a steel surface, virus can stay for a particular time. So this transmission actually mainly depends on the type of the surface. Incubation time of this virus in a human body mainly 2 to 14 days. So now the source of this virus. The virus was first detected in the Wuhan city of Hubei province of China, and it's linked to the animal market. So there are a lot of controversy going on with this for the source of this COVID-19, but definitely its source is related to the animal. The SARS-CoV-2 virus is the 7th member of the coronavirus family. Among the family members, SARS-CoV, MERS-CoV, and SARS-CoV-2 cause this severe disease. Apart from this, the other 4 members are not that much as as that much infectious to the human. The recent study showed that the bat-derived coronavirus shows 88% as identity. So bat could be a source of coronavirus. Or another study showed that pangolin-derived coronavirus exhibit some similarities with SARS-CoV-2. So pangolin could be another source of this virus. But according to the Nature Medicine March edition, it is clearly showed that the SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. The source of virus is definitely natural. How the cells, now the question arises, how the cells get in to the human body. So the virus top surface is actually consists of spike protein that is called S protein. S protein contains 2 subunits, S1 and S2. The S1 has the receptor binding domain which can bind with human angiotensin receptor 2 as a receptor. And actually this there is a complex formed by binding with the virus receptor binding domain and human ACE2 receptor. By forming this complex, the virus cells can get into human body. So now the question arises that why the virus is so infectious. The crystal structure of SARS-CoV-2 receptor binding domain is showed that there is a significant structural difference between the receptor binding domain of SARS-CoV-2 and SARS-CoV. Due to that structural differences, the SARS-CoV-2 receptor binding domain has the significantly higher affinity to bind with the human ACE2 receptor. That is the reason the virus is so infectious. The coronaviral genome encodes 4 structural proteins, major proteins, which is spike protein, nucleocapsid protein, membrane protein, and envelope protein. The spike protein is mainly responsible for the viral entry to the human body. So now, the diagnosis of this COVID-19, how we can say one person is COVID positive, another one is virus negative. The process or by the technique by which we can measure the viral genome traces in human body is the qRT-PCR. So what is qRT-PCR? Is the technique by which we can amplify a particular segment of a DNA to 1 million copies. There are 3 components of this technique, the real-time PCR. One is a segment of the DNA, which is called the template. Another one is the primers. Other one is the DNA polymerase enzyme. By the DNA polymerase enzyme, the primer binds with the DNA segment and form 1 million copies of this particular DNA segment. So for the diagnosis of COVID-19, as a template, we consider multiple viral genes. Amongst them, ORF1b and N are important. ORF1b is a protein which plays an important role in viral pathogenesis. Other proteins we can use as a marker of the template DNA, that is the RNA-dependent RNA polymerase, spike protein, nucleocapsid gene. Some cases, the E gene PCR positive also considered the person is the COVID positive. So possible therapeutic approaches. Early diagnosis, quarantine, and supportive treatments are essential to cure patients. Antiviral agents, chloroquine and hydroxychloroquine, corticosteroids, antibodies, convalescent plasma transfusion, and vaccines are the possible therapeutic approach to fight against this deadly virus. Multiple companies in different countries are developing vaccines in different approaches. Hopefully, the vaccine will be available to us end of this year or early 2021. But I'm not going to details in the vaccine development. Today, I'm going to mainly discuss about the current therapeutic approaches which are taken to fight with this deadly virus. Among them is very important is the antiviral compound remdesivir. Remdesivir is originally developed for the treatment of Ebola virus. In cell culture, an animal model showed that remdesivir can block the replication of variety of coronavirus. In a mouse model of SARS-CoV pathogenesis, after application of this drug, it showed the significantly increment of the pulmonary function, increase the severe lung pathology, and reduce the viral load. The remdesivir, currently in US, remdesivir is authorized only for the patient who is severely ill and who is already admitted to the hospital. And the drug must be administered intravenously. So when we are talking about a drug, we should know how the drug is working. The data showed that the main target of this drug is viral RNA-dependent RNA polymerase enzyme. Basically, after diffusing this drug to human body, it actually by a phosphorylation form an active analog that is nucleotide triphosphate. And this viral RNA, a viral enzyme, RNA-dependent RNA polymerase actually utilize this NTP and stops the to add more RNA subunit to the growing RNA chain. Basically, after adding the remdesivir, the enzyme stops being able to add more RNA subunits, and it's finally halt the genome replication. So the next drug is chloroquine, hydroxychloroquine. It's most controversial drug right now, though in several cases till now this drug is given. So chloroquine is a very well-known anti-malarial and autoimmune disease drug that has also the antiviral property. So chloroquine is known to block the viral infection by increasing endosomal pH, which is required for virus cell infusion. Is also interfere with the glycosylation of the cellular receptor or SARS-CoV. Hydroxychloroquine is the analog of chloroquine, and which is actually more potent to fight against this virus. So after this disease onset, several cytokines are upregulated as a part of hyperactive immune response. Among them, cytokine IL-6, IL-10 are important. So this both chloroquine and hydroxychloroquine has the immunomodulatory property by which it can reduce this overactive immune response. So now I am to start this main focus topic of these current days, the convalescent plasma therapy. So currently, what is that? Currently, there are no vaccines, monoclonal antibodies, or drugs are available for the SARS-CoV, although many are in rapid development and some may be available in a short time. So that is the case when convalescent plasma therapy is actually strategy to give the passive immunization to reduce the infectious disease, and its therapy actually is given since the early 20th century. The major target of this therapy is to neutralize the pathogen. This convalescent plasma therapy has been considered as an emergency therapy in several pandemics before. Among them, Spanish flu, Ebola virus are important. So, history of this therapy. Concept of convalescent plasma therapy infusion was first established in 1880, where it was shown that the immunity against diphtheria is relied on the existing antibodies in blood. Basically, in the blood from animals, they intentionally immunized with non-lethal dose of toxin, which is basically a vaccine, that could be transferred to the animal suffering from active infections. In early 1950s, purification and concentration of the immunoglobulin from healthy donors or recovered patients provide an option to treat the serious infectious disease. The use of convalescent plasma therapy was widely used in different outbreaks as the quick therapeutic option given the lack of effective medications or vaccines. Currently, this therapy used in the patient with COVID-19 demonstrate a definitely reduction of viral load and clinical condition. So from Spanish flu influenza to current pandemic caused by SARS-CoV-2, it has been observed that the use of convalescent plasma therapy significantly reduced the case fatality rate. So what is the basic of this therapy? This is the donor, who is the person who is recovered from COVID-19. The blood is collected from this person, and from the blood, the plasma is collected. In the plasma, there are the neutralizing antibodies are present, and this plasma is transfused to the human, the person who is actively infected with the virus. So now, plasma collection for this therapy. The convalescent plasma donor must undergo the standard blood donation assessment test. Currently, these donors' age are considering between 18 to 65 years old and without no infectious symptoms, and the person should be COVID-19 test negative after 14 days of the recovery. The test must be repeated after 48 hours of the blood donation. The apheresis is the technique by which plasma is collected from the blood. Basically, the whole blood undergo the centrifugation and the plasma is collected from there. So generally, on a single blood donation, 400 to 800 ml plasma can be collected. And after the collection, plasma should be allocated to 50 ml per unit. And within 24 hours of the plasma collection, it should be frozen for the future use. The dose of this therapy, currently there is no standard transfusion dose for convalescent plasma, though in US, is following this convalescent plasma is giving to the person is 3.2 ml per kg of body weight. So if the patient's body weight is 80 kg, the person should receive approximately 250 ml plasma per day, and it's needed 2 days of the plasma therapy, plasma transfusion. So this is the reason while the plasma was collected, it should be aliquoted in 250 ml per unit. The composition of this plasma therapy, the convalescent plasma consists of the neutralizing antibody. This is IgG SARS-CoV-2, IgG SARS-CoV-2. Apart from the neutralizing antibody, it has also non-neutralizing antibodies also from the donor. And these exert a protective effect against this viral disease. After the viral infection onset, there are several cytokines are upregulated, as I discussed before. This is a part of overactive immune response, and sometimes it leads to cytokine storm. This is a very well-known term in this viral disease. The high fever of the patient is mainly due to this cytokine storm or the overactive immune response of his own body. So these neutralizing antibody or non-neutralizing antibodies, by its immunomodulatory effect, this can reduce this hyperactive immune response. So now the antiviral mechanism. Neutralizing antibodies are very crucial in the viral clearance and also give protection against the viral disease. The efficacy of this therapy is mainly depends on the concentration of the neutralizing antibodies in the plasma from recovered donor. In SARS-CoV, it was discovered that the neutralizing antibody actually binds with the receptor binding domain of the spike protein, and thus it actually inhibiting the viral entry to the human body. Finally, it inhibits the viral amplifications. Studies showed that SARS-CoV specific antibody bind with a COVID-19 receptor binding domain, and this antibody did not show any competition with ACE2 for the binding to COVID-19 receptor binding domain. The receptor binding domain of the COVID-19 varies broadly from SARS-CoV at the C-terminus residue. Although this difference does not enable COVID-19 to bind ACE2 receptor, does influence the cross-reactivity of the neutralizing antibody. Interestingly, the plasma from the recovered patient shows a significant variation in the neutralizing antibody titers. 30% of the patient didn't show any significant amount of high neutralizing antibody titer in their plasma. So in plasma, addition of the neutralizing antibody, there are also protective antibodies, including IgG, IgM. The non-neutralizing antibody bind to the virus. Maybe it do not affect to the replication capacity, must contribute to the prophylaxis or the recovery improvement. So what is the term called prophylaxis? Prophylaxis is the action is taken to taken the protection against the viral disease when the disease is not onset. So a recovered donor from the COVID-19 infection had SARS-CoV-2 specific antibody titers ranging between 1.8 to 16.2 and non-neutralizing antibody titers between 80 to 480. This antibody titer is generally measured by ELISA. It's a well-known technique to measure the enzymatic activity. So after the transfusion of this convalescent plasma, the titer of IgG and IgM in the recipients is increased in a time-dependent manner. Moreover, the presence of the neutralizing antibody in the recipient play an important role in reduce the viral load and this restriction of the viral infection. The titer of the neutralizing antibody in the infected patient didn't show any positive titer in the first 10 days of the disease onset. It significantly increased the antibody titer in 10 to 15 days after the disease onset, and then it remains stable. So risk, the human plasma transfusion is a regular event in all the modern hospitals. The difference between normal plasma and the SARS-CoV-2 plasma is only the presence of the antibodies against SARS-CoV-2. Donors should satisfy all the standard blood donation the criteria according to the federal government and state. This risk of transfusible infection is nowadays very low in US. So there are also some non-infectious hazards of the transfusion. Among them, some are the allergic transfusion reaction, transfusion-associated circulatory overload, transfusion-related acute lung injury, cardiorespiratory disease, renal impairment are important. Interestingly, this virus is not transmitted via blood like HIV or the hepatitis B. Only 1% of the infected patients shows the viral traces in their blood. So the plasma transfusion in this case is completely safe. In summary, the key factor of the succession rate of the convalescent plasma therapy depends on the donation of the plasma by people who have recovered from the COVID-19 and have high concentration of SARS-CoV-2 antibodies. The safe preparation of plasma is also an important factor. Many donors are needed because only a plasma donation from one patient is mainly can serve only few patients. And in the US, the Red Cross in collaboration with FDA has published a call for donors. In each country will need to do the similar thing if this therapy is to be used to treat the most severely ill patients. An additional use for anti-SARS-CoV-2 antibodies isolated from the plasma is the development of a concentrated therapy known as hyperimmune globulin, in which antibodies from many donors are pooled. The current pandemic provides the greatest opportunity yet to develop convalescent plasma therapy into viable treatment option for COVID-19 while a vaccine is developed. In conclusion, this global pandemic and the desperate need for effective treatment for the patients with severe and critical COVID-19 is driving both basic and clinical research at an unprecedented rate. The human plasma from recovered COVID-19 patient is projected to be a safe and potentially effective therapy for treatment. In the acknowledgement, I want to acknowledge my current PI, Dr. Sumanta Goswami. He is Associate Professor and Chair of our department, and he is also Associate Professor in Albert Einstein College of Medicine. Dr. Maya Octe, he is a very well-known pathologist and professor of Albert Einstein College of Medicine. Actually, she provides the cancer patient FLA sample to our lab. And lastly, Dr. Barshanjit Mojumdar, he is a professor of Cleveland State University. He is now is a very well-known writer in Bengali dailies about this coronavirus viral pandemic. I discussed with him a couple of times while I was writing this presentation. These are the references that I used for the presentation. And I would like to conclude my presentation with a very recent quote of a well-known American physician and immunologist, Dr. Anthony Fauci, that the coronavirus is just took over the planet. And we all have to work hard, and we have to work the fight against this virus, maybe for a long time. So that is my presentation. So now the questions. I'm ready to accept the questions.
Thank you very much, Dr. Bandyopadhyay, for your lucid deliberation on therapeutic approach, how to combat COVID-19. So we have a lot of questions. We cannot take all. Maybe we take 3 or 4 questions for now. So, Madam, what is the main difference between Ebola virus affected cell and SARS-CoV-2 affected cell?
The Ebola virus is a more deadly virus, though it's less infectious compared to the SARS-CoV. Definitely. Is almost 8%. Its mortality rate is around 8 to 9%, whereas the SARS-CoV is 2 to 3%, 2%. So it's more infectious. SARS-CoV definitely more infectious, but less death rate compared to the Ebola virus. That I can tell.
So as you were sitting in New York, what is the situation there now?
This is, yeah. Now I can tell is we are in a little better situation compared to 3 to 4 weeks back. It was in New York city states, the coronavirus count was approximately 350,000. What is a count of a whole countries generally? So it definitely, we are in a better situation compared to before. As of Andrew Cuomo's last speech, I can tell you it's now reduced to 65 deaths, and it's the new infection is around to 700, 800 per day, which is compared to the before, it was 3,000 to 4,000. So a better state, I can tell you.
Okay, can plasma therapy be used extensively?
The plasma therapy is some cases, it's definitely reduce the case fatality rate. But as I mentioned, that there are some difficulties of this therapy also, some non-transfusion hazard also, that the lung injury, cardiovascular arrest, and the donor's health condition, the patient's health conditions also depends. So in US, definitely it helps. And in the New York perspective, I can tell, yeah, it's helped in the plasma therapy. In India also now they started in few cases, but they definitely started in India, Delhi, as of the last report.
So how COVID is spreading from bat or pangolin? Is it through droplets or to eat these animals or any other reason?
As definitely not the droplet. Droplet is transmission transmitted by the human droplet, but by eating, maybe is the bat or from pangolin, the virus is infected to the human, maybe by eating. Because they are in China, that province, they have the live animal market, and they have various this wildlife, wild animal eating habit. So, and before SARS-CoV also actually was infected by this bat before. So it's definitely related to that live market.
There are many questions from Arnob Banerjee. I am selecting one. Why parasitic drug like ivermectin is used in the treatment of COVID-19? What is the relationship between parasitic nature and COVID-19?
Honestly, I don't have much idea, but definitely the parasitic drug is not that effective in certain cases that right? These chloroquine, the hydroxychloroquine cause the acute cardiorespiratory problem to the patients.
Is that still using in USA?
No, right now FDA is completely stopped to use that drug. Initially, first 1 to 2 months it used, but currently it's stopped. using this drug.
So there are plenty of thank you messages. You can also check for your video presentation.
Sure. When you actually first requested me to talk about coronavirus, I just wanted to tell you that initially I thought what I'm going to write or what I'm going to discuss, because I'm not in this field. So I'm completely different field, but though now it's current pandemic situation, and I'm in the New York City, which is New York City going to a very rough phase. So that's why I thought, why not?
None of our 5 previous speakers have elucidated therapeutic approach in details, right? What you have said. So you are the unique one. So you have fitted well. You don't have to fear. And
thank you.
So many
thank you. Thank you so much.
And as CP therapy has a limited use in terms of the number of recipients, can plasma be collected from asymptomatic patients also? It might increase the number of recipients. Is it possible and safe?
No, because asymptomatic patient, we have to measure that, as I mentioned in my presentation, the person should be COVID-19 test negative after 14 days of recovery. So asymptomatic patient definitely show the test positive, right? So how can you transfer the plasma from an asymptomatic patient? So the person should be COVID-19 test negative. And this test should be again repeated after 48 hours of the blood donations also. So as of my knowledge, the plasma cannot be transferred from the asymptomatic patient.
So one direct question from the research field that you has shown us in the first 5 slides. Does the hormones enhance breast cancer?
Yes, obviously.
There are plenty of questions. I am limiting to 1 or 2 now. Let's take some questions from a student. How did the success of treatments varies among patients?
Sorry, say again the question.
How did the success of treatments varies among patients?
Okay, so this actually depends on various factors. That the patient health conditions also depends. And another discussion I can tell that
Is obesity the issue?
No, not really. Obesity might be an issue. And another question I can tell you that in this regard, that why the one person has very severe symptom, the another person with the same virus has less symptom. What is the reason? Because of the genetic formation. Gene analysis is also depends the person to person. That is also very important to this therapy also. That how this therapy it actually varies from each person to person and effective. So this is a very unique term. Is now a single nucleotide polymorphism. This is also important. That this difference actually varies the how infection is. This symptoms varies from person to person, actually.
So,
so that Salam, means that the treatments also varies from person to person. Yeah, yes. Go ahead.
We can take the who has raised hands. Please connect with them.
Okay, we have Dr. Parashar Banerjee. Dr. Banerjee, are you getting us? Dr. Parashar Banerjee?
Maybe the next one.
And again, Dr. Probir Karar is with us. Dr. Karar?
Hello.
Dr. Karar, yes.
Yes.
Introduce yourself and place your question.
Okay, thank you. Myself is Dr. Prabir Karar, Associate Professor of Economics, Bhairav Ganguly College. Ma'am, thank you. We have ever had, actually. My point is that why don't the government of different countries of the world, you see every country are now affected, why don't the government of each and every country is specializing the institute, particularly medical research institute and the hospitals to go for the plasma therapy? Because as you said earlier that it is safe till we have a vaccine for COVID-19.
Yeah,
we can strongly go for this plasma therapy, and in India there is a report that the people are ready to give their plasma who have recovered from acute COVID-19 cases. So why not we have a donor bank for plasma and can treat the most serious patient who are in ICU or ventilator? So why government are hesitating in that situation? I don't understand. Please.
Yeah, absolutely. So maybe there are few factors that are involving. That is, I told that the safe preparation of plasma from the blood is very important factor, and the many donors are needed. So there are the people awareness also very important.
Yeah, that's true.
That lot of people needs to come out and they are ready to give their plasma donations. So that is the people awareness also important. And that is now here in New York, that is what started that the plasma therapy in first in I think on the March end or April. But that definitely a lot of people need to donate their blood, because from one person it can be delivered. It can actually solve the problem only very few patients. So we need a lot of plasma, and the plasma should be separated, and the blood transfusion, all the methods should be considered very carefully for the plasma transfusion. So in such a large number of patients is also very, I would say, is not critical, is very important factor.
As a case in point, the state of Kerala in India, they have had a very good track record by just curing or preventing COVID-19. And as far as my knowledge goes, that they have used plasma therapy intensively in their state and dropped the death rate to a bare minimum,
So you need to, track record also an important factor. Track record, you should know that which patient is recovered and the factors that I mentioned, that is also very important. So now I saw that news that Delhi also. They started to use the plasma therapy. This is not a long time solution for the current situation. This is a definitely important therapeutic approach to fight against this virus.
Yes. Thank you very much. Thank you very much.
Dr. Parashar Banerjee, are you getting us? Please unmute yourself. Dr. Banerjee, if you are available.
Yeah, I am unmute.
No, not you, ma'am.
Oh, okay.
Parashar Banerjee.
Oh.
Okay, Dr. Bhadra, yes, you can carry on. I think there is no more participant to raise any questions.
2 or 3 questions in the chat box. What are RH limitation blotting techniques?
This is the RH factor test is the standard transfusion process. You're talking about the RH factor matching, right? For the plasma transfusion. This is the basically the same standard blood donation matching for the RH factor. There's A positive, AO blood group.
Okay, one last question. Does PCOD is a symptom for breast cancer? Could you please give some inputs on this?
PCOD is related to the hormonal imbalance and estrogen, and yeah, definitely it has some effect with the breast cancer, but yeah, right now I cannot tell you in details that how it's related to the promotion and progression of the breast cancer. So yeah, I can tell you later by reading and all these things by email. But definitely, yeah, because there is a hormone linked to the estrogen positive, ER positive, ER negative. Those are related to the types of breast cancer. So definitely, yes, the hormones are responsible.
One major concern, why mortality rate is lower in younger than older?
This is a good question. That's just the last week I was discussing with my PI regarding this. He is also working as a team of in a part of CDC in New York. So I was discussing with him that in India, maybe the mortality rate is mainly due to the more young populations. But there is no scientific description that the young population has the less fatality rate. Means the statistics shows that the young population case fatality rate is low, but there are some cases in New York that's 27 years old, is very healthy doctor, is now almost supposed to death. He just came back from the ICU of the hospital. So there is no scientific evidence, proper scientific evidence that the young persons has a less fatality rate. You can say the statistics in South Korea, India has the less mortality rate compared to European American country due to the age related factor.
Dr. Banerjee, we have one participant observing our YouTube live demonstration, live streaming. Devojyoti Pradhan. Devojyoti Pradhan is there, who has a question. Do you think natural herbs can boost our immunity to get rid of from coronavirus?
I don't have much idea about that. I read a few articles, but is there any scientific method that I have no idea about that. Immunity boost up is definitely a way to fight against this virus, but I have no idea about the natural herb that can fight against this virus.
Another question from the same individual, that is what would be the affordable cost involvement in plasma therapy treatment?
It's not much as of my knowledge, because we are getting the plasma from the patient, from the recovered donor, and it is apheresis is the process by which plasma is generated, is processed from the blood. So it's a regular plasma processing process, apheresis. So it's not a very expensive way of the treatment. It's not very expensive. I cannot tell you the exact amount of money that how much is required, but it's not that much expensive.
Okay, thank you, ma'am. Dr. Bhadra, over to you now.
So one last question as we are heading towards the end of the session. It is speculated that in children, the ACE2 receptor are not developed, which makes children carriers without infection. Is it true?
Yeah, absolutely. To some extent, definitely yes. And maybe you heard about the recent news in New York and New Jersey also, that in some kids, before 15 years, around 8 to 17, 18 years, there are severe symptoms of this COVID-19 related disease. This is not exactly the COVID-19, but it's related to that. This is called the Kawasaki symptom, that the kids has more high, kids' immune system is very active. It caused by this cytokine storm like situation is happened to the kids. So yeah, that's how the kids are related to this means disease. It's not very much. The kids count are not that much, but definitely kids are also affected by this.
So there are a lot of questions. I personally request you to see in the chat box later.
Okay, sure, I'm sure. Yeah.
So from the organizer, as we are at the end of the session, I formally thank you once again for accepting our invitation and give a fruitful deliberation apart from your doctoral, postdoctoral and scientist work, and then COVID-19 in therapeutic approach. We sincerely thank you for all staffs, and thank you very much, Gargi.
Yeah. Thank you very much, all of you too, to selecting me for this participation, and I'm very much thankful to all of you.
Thank you very much.
Oh, sure.
So as we are near the end of our two-day webinar, I will take this opportunity to convey my vote of thanks to all the people who made this event possible. I would like to thank our Vice Principal, Dr. Sanjit Kumar Das, my colleague in organizing committee, the IQAC coordinator, Dr. Shamoli Dewan, and all the members of the IQAC who have made a driving force behind the success of this webinar. This would not have been possible without our special guest, Professor Bashok Choudhury, the Honorable Vice Chancellor of our affiliating university, West Bengal State University, and our speaker for keynote address, Professor Dubojyoti Chattopadhyay, and distinguished resource persons starting from Nirmallo Dasgupta to Sumita Goswami to today's Riya Bose, Tanmoy Chakraborty, and finally Dr. Gargi Bandyopadhyay. Dr. Abu Salam, who ensured that the technicalities of running a webinar were looked very fruitfully. Last but not the least, it would not have been possible without our esteemed participants. I thank research scholars, teachers, students from different parts of our country who stayed with us for 2 days, showed patience during technical glitches, made the question answer session interesting through their questions and comments, making this webinar a forum for discussion on the most relevant topic of this time, COVID-19. Despite the current situation being so dismal, I would like to believe that there is light at the end of the tunnel. After this 2-day webinar, presentations made by the resource persons who have got a realistic picture regarding the research being done in the development of COVID-19 vaccines, and we may begin to hope that later, if not sooner, immunization will help us to live with the coronavirus rather than living in fear of it. For the time being, we can do our bit by taking precautions like using hand rub, washing hands frequently, wearing masks, and maintaining social distancing more. So now when we are gradually returning to the normal rhythm of life, stay safe, take good care of yourself. Good night. Thank you all. I finally like to request our Vice Principal, Dr. Sanjit Kumar Das, to give his concludatory remarks. Over to you, sir.
Thank you, Dr. Bhadra, and good afternoon, everybody. First of all, I express my heartiest thanks to the participants and viewers at this final stage of 2 days international webinar organized by the Department of Physics and the IQAC of Bhairab College. And in this valedictory session, I express my gratitude and thanks to the keynote speaker, Professor Dubojyoti Chattopadhyay, the Honorable Vice Chancellor of Sister Nivedita University. And I also express my gratitude to the 5 distinguished speakers, Dr. Nirmala Dasgupta, Dr. Sumita Goswami, Dr. Riya Bose, Dr. Tanmoy Chakraborty, and Dr. Gargi Banerjee for their presentation. Staying from different part of the world, they have discussed the present scenario of the COVID-19 and the possible measures to counter the solution. And from the point of view of the scientists, they have shared the information of the laboratories working for the benefit of the mankind at this moment. And they have provided a lot of valuable information and insight, and we have enriched to face the challenges in this pandemic period. And I am also thankful to Dr. Bhadra and his associates of the department, and Dr. Deon and our associates of the IQAC cell, and also Dr. Abdul Salam for his technical support that make the possibility of this seminar a grand success. You know that with the attack of COVID-19, the socioeconomic structure of the entire world has been cracked, and we don't know how long it will take time to recover the situation. But we can definitely say that there will be some changes in the lifestyle, in the economic structure, in the occupation pattern, and different aspects of life. Particularly in the higher education sector, we have to face a lot of challenges in future, and the education system, examination system, evaluation system will be changed, and we have to adapt and accept these challenges. And we have to change our attitude. Simple change in the attitude and perception can make a huge difference. And as a teaching community, we have to provide all sort of support and information to the students who are in really in great danger. They cannot come to the college, they cannot meet the students and teachers. So we have to support them all time, and we have to counsel them so that they could solve the problems. And in the end, I would like to mention the statement of our Honorable Vice Chancellor, Sir, Professor Basub Choudhury. He made in this inaugural session make a comment that in spite of these obstacles, in spite of these hazards, we have to move with full potential, with self-confidence and our wisdom. Depending on our wisdom, we can make these challenges and we can become successful. I hope you have enjoyed all these sessions, and also hope that we will provide our support and cooperation in the near future when we will execute any other forthcoming academic endeavor. So stay safe, stay home. I again thank all of you for making this webinar a grand success. Thank you.
Thank you, Dr. Dash. One general comment for the participants. Now the feedback form is open, and I have several times delivered you in the confirmation emails. Now the feedback form is open. It is open till 24th June at 5 p.m. So please fill it up, and whoever is not registered yet in the MOOCs BGC library, moocs.bgclibrary.in, get the participation certificate. Please hurry and do register yourself in the in that learning management system app. It is mandatory to get it, the participation certificate. And finally, I would like
Can you hear?
Dr. Bhadra, I think there is some
international on 27th Ranjan Thakur. Sorry for the interruption. It is available in the Bhairav Ganguly College website. And finally, I would like to thank all for making this event a grand success. Over to you, Dr. Salam. And
Dr. Bhadra, please mention the next webinar.
It is going to held on 27th of this month, June, where Soikat Ranjan Thakur from, again from US. You can see the poster like here. Yes, the Soikat Ranjan Thakur. He will be connect us from San Diego, US again. And for the students also, particularly, this event is made for you only. please do register yourself so that you can enriched by your understanding and knowledge. Thank you, Dr. Salam. So this is all for now. We can leave.
Okay, thank you very much to all of you for a grand success of this seminar at international level. Thank you very much. We are ending this session.